Good evening, all the participants. I welcome all of you for the webinar series on sheep and goats. Topic of today's webinar is pathogenesis and pathology of contagious ecthyma and the other poxviridae in sheep and goats. Speaker is Dr. Nawab Nasirudullah, Professor of Pathology, working at SKUAST Jammu. I welcome both the speaker and special guest, Dr. Nawab Nasirudullah sir and Dr. Joshbina Tapa. Welcome both of you.
Thank you, sir.
I welcome all the four permanent invitees of these webinars, Dr. Umesh Chandra Sharma, President Veterinary Council of India, Dr. Arun Kumar Tomar, President ISHGPU, Dr. Chanda Nimbukar, Director Animal Husband Division NARI, Dr. GV Ramanjanelu, Executive Director CSA. Respected participants, the main aim of organizing these webinars is promoting zero grazing of sheep and goats. The objective is capacity building of field veterinarians by dissemination and exchange of knowledge. For this, we have been conducting webinars since last April 2021, and today's webinar is the 87th webinar. And for exchange of knowledge, we are maintaining WhatsApp groups also for daily updates. There are more than 6,000 veterinarians representing from almost all the states of India. Few of them are from African countries also, who have one YouTube channel with the name of Sathyazerograzing, and it is open for all. There are more than 220 videos available in this YouTube channel, which are particularly on sheep and goat sector. We are associated with ISHGPU, that is Indian Society for Sheep and Goat Production and Utilization, NARI, Nimbukar Agriculture Research Institute, Maharashtra, CSA, Center for Sustainable Agriculture, Hyderabad, and KVA, Karnataka Veterinary Association, Karnataka. And these webinars have been promoted by IVA Lady Wing, Jharkhand, Pashudan Prahari, Blue Cross Welfare Foundation, Like Weights of Odisha, and Omega Laboratories of Maharashtra. Now I will introduce today's speaker, sir, Dr. Nawab Nasirudullah, MVSc PhD, presently working as Professor and Head at the Division of Veterinary Pathology, Faculty of Veterinary Sciences and Animal Husbandry. The university is SKUAST, that is Sher-e-Kashmir University of Agricultural Sciences and Technology of Jammu. And he has completed his BVSc in 1997 and MVSc in 2000 and PhD in 2014. All are from Assam Agriculture University only. So he is alumni of College of Veterinary Science, Assam Agriculture University, Guwahati, Assam. So in 2002, he joined as assistant professor in Kashmir, that is in the same university, SKUAST. And from August 2008, he is working as associate professor in the same university, but at Jammu. And from 2015, he is working as professor at SKUST Jammu. His area of specialization is veterinary pathology, research interest, pathological conditions affecting the captive wild carnivores, and paratuberculosis in small ruminants, and parapox virus, that is contagious ecthyma, parapox virus infection in goats, and lungworm infection in domestic and wild ruminants. So under his advisory, 2 students have completed their PhD and 3 have completed MVSc, and 1 PhD student is pursuing his research under his guidance and advisory, and 1 MVSc student also is pursuing his research. And student research includes Pasteurella multocida infection in domestic animals and birds, and AV pox virus infection in birds, Capripox virus infection in small ruminants, and avian orthoavilovirus infection in poultry and pigeon. So far, he has completed 11 projects, and 8 are funded by SKUST Kashmir, and he has completed them as principal investigator and co-principal investigator. And 1 project funded by SKUST Jammu, it is completed as co-principal investigator, and 1 funded by RKVY, completed as co-PI, and 1 DBT funded societal project completed as co-principal investigator. Total number of publications at his credit are more than 77, and there are 31 manuals published in different books, manuals, and monographs. and extension trainings associated with are 4 as course coordinator. He has received 4 awards so far. One is University Merit Scholarship during his undergraduate and postgraduate from Assam Agriculture University, Assam. And one Krushi Vigyan Gaurav Upadhi Award by ICR Bharatiya Krishi Anusandhan Patrika in 2016-17. And one Best Poster Award for Research Paper in International Congress and Best Paper in International Conference. This is in brief about today's speaker. Now I request today's special guest, Dr. Joshbina Thapa, who is a veterinary officer and in charge of Bonpala Sheep Breeding and Conservation Farm. She is working as Department of Animal Husbandry and Veterinary Sciences, Government of Sikkim. Madam, I request you to kindly address the participants. Over to you, madam.
Okay. Good evening, everyone. First of all, I would like to thank Dr. Ashok Kumar Valupadasu and the organizing committee of Sathyazerograzing for having me here. I feel honored and privileged to be part of this national webinar. Myself, Dr. Josbina Thapa, and I am from Sikkim. I have completed my BVSc and AH from the College of Veterinary Sciences and Animal Husbandry in the year 2018. And currently, I'm working as veterinary officer, in charge of Banpala Sheep Conservation and Breeding Farm for the past 1.5 years under the Department of AH and VS, Government of Sikkim. So that is the brief introduction about myself. So before moving on with the main topic of today's webinar, I would like to speak few words on the importance of sheep and goat. As we all know that goat and sheep husbandry, it plays an important role in Indian economy, especially in India. It plays an important role in the lives of the weaker section of the society because it contributes, it provides livelihood to the weaker section of the society, and it provides livelihood to almost 2/3 of the rural community. And in India, it is also described as mortgage lifter of India. And the sheep and goat sector, it contributes greatly to the agrarian economy, where especially in the areas where crop and dairy farming is not economical. And as we all know that sheep and goats being small in size, it is easily reared by women and children also as compared to dairy farming. And nowadays, it's getting more importance because the initial investment for starting this farming is less compared to dairy farming, and also it can thrive in wide variety of vegetation as well as in wide variety of agro-climatic condition. And also this sheep and goat, they are less susceptible to diseases, and it promotes job opportunity also, as well as it helps in elevating the poverty. So, and this nowadays in Sikkim, like unemployed youths, they are taking this sheep and goat sector as entrepreneurship, and this helps in making them self-reliant as well as self-sufficient. And they not only it not only gives livelihood to themselves, but it also helps others to get a job in the enterprise. So that's all. Now I thank you so much, and I would like to hand over to Dr. Ashok. Thank you.
Thank you, madam. Thank you very much for accepting the invitation and also for participating as special guest. Now I request today's speaker, Dr. Nawab Nasirudullah sir, to kindly start his presentation. Over to you, sir.
Thank you, Dr. Ashok Velupadasu. And then I also thank Zerograzing for giving me this opportunity. And I also thank Dr. S. who gave a very good introduction about the importance of sheep and goat farming. And as already know, everyone knows sheep and goat is considered as poor man's cow. The stress is particularly in certain select states and a certain sector of our farmers, they rely heavily on sheep and goat. So without much ado, I'll skip directly to our topic today. I'll be sharing my screen. The presentation is visible to everyone now.
Yes, sir.
Is all right, sir?
Sir, please continue, sir.
Okay, so I thank Sathyazerograzing and all the people, all the esteemed scientists and experts and farmers.
Sir, please off your video, sir. So that. Yes, please continue.
Okay. So I thank everyone today present in this August gathering. This is a national webinar, and I believe there are many experts, many scientists, and some of them may be prospective farmers also. And I hope my topic of discussion today will benefit them very much. So my topic today is pathogenesis and pathology of contagious ecthyma and other pox variety in sheep and goats. Like being a man of pathology, I really put stress on the word pathogenesis and pathology. But then again, I was cautioned not to speak too much about pathogenesis because that might, cause a little deviation of interest for most of the general public. But then again, I always consider our esteemed guests to be well versed with pathogenesis as well as in pathology. And the things that I am going to discuss today is just a refresh of our the experience that we already hold in case of sheep and goat. So I have been introduced. I'm a professor in veterinary pathology. I work in Sherry Kashmir University in Jammu. And some of these works I have done myself, and some of the works have been done by our students in pathology department. And I will be sharing my presentation today as well as part of our research experiences. So the pox variety is a large group of viruses. It is a family of double-stranded DNA viruses, and it affects many humans, vertebrates, and even arthropods as natural hosts. And there are different species and genera. Again, these numbers they always keep varying. There is a association called as the ICTV, or International Committee for Taxonomy of Viruses, and they review number of species and genera, and they keep on scheduling and rescheduling these different groups of those viruses, and therefore I have put them on in red now. But basically, there are two subfamilies. One is called as the pox variety, Entomopox variety, and another is called pox variety. Okay. So the Entomopox variety is of much less interest for us because they usually affect insects. And we have the Caudopox variety and the Orthopox virus. Okay. So here today, our interest is here in a group of our genus of virus. We call it as the Orthopox virus and the Capipox virus. This is the general homology or genetic relationship between the different genuses and species of viruses. If you see here, this is the sheeppox virus, then we have the LSD virus, then vaccinia virus, then ORF virus. So all these viruses, how much they are related to each other. So this is a general representation, but it always comes down to a very common root. And there are certain literature which discusses about the origin of the different pox viruses, how they have evolved, and how they have separated and made into distinct class. So of course, these topics are beyond the scope of today's presentation. So we'll skip some of the slides. Like we have already discussed. These are the different viruses and hosts. Caudopox variety, I already discussed. They usually affect different insects. If you see here, this is Hymenoptera, Coleoptera, Diptera. So they refer to different groups of insects. Lepidoptera here are pox viruses that causes diseases or pox diseases in case of butterflies. And again, we have certain groups that cause pox infection in case of mammals. Then every pox virus, there is a different class of pox viruses that affects the birds. And so basically they all causes, some type of skin lesions. Some of them are restricted to the skin lesions alone, and some of them may have certain, reflections in a systemic way. So we'll be discussing this in part. Here, see, we have already shown some of the representative, species of pox viruses. Here, for example, this first slide, this shows the lumpy skin disease, which are presently emerging disease in India, and it has really caused havoc in certain states in the northern and the northeastern states also. And this is pox virus infection in hares and rabbits. This is pox virus infection in case of deers. Then we have any pox virus, then we have sweet pox virus, we have any pox viruses, and then we have got sheep pox virus. So particularly, all pox viruses invariably they affect the skin, and they form nodules and disseminated rashes. Then again, there are certain pox viruses, for example, the sheep and goat pox viruses. They may also have some additional predilective. For example, they may have a systemic infection, and they may cause lesions in the lungs and other visceral organs. These pox viruses have been known to humankind since thousands of years back. So traces of smallpox viruses have been, identified even in the mummies of Pharaoh Ramses V, and he's you can see the picture here. I think you can appreciate the reasons here. So these pox viruses have been affecting humans and animals since thousands of years back. And there have been, certain written documentations about pox viruses also. For example, in case of smallpox, the from China, and that was, around the 4th century CE or after butterflies. Then in India, we have documentation about smallpox from the 7th century. And in Asia Minor, we have, documentation since 10th century CE, and likewise. Now, these pox viruses have been responsible for, modeling and remodeling human evolution, as well as our, even they have shuffled or rearranged the pantheon of the gods also. Now, in India particularly, we have a specific goddess or mata, we call it Sitla Mata. And she has been endowed with the sole responsibility of healing people from poxes and sores and pustules and other diseases, particularly diarrheal diseases. And she has also been linked with smallpox. So in India, when at the times of the smallpox epidemics, she had been regarded and venerated, and her vehicle here you can see is a donkey, and in one hand she will be carrying a broom, a jaru. So the broom indicates she will be dusting of the dust and other dust particles, and also she would be providing some kind of a relief to the patients, smallpox patients. And also she will be carrying a pot where she used to carry some emollients or certain medicines, particularly a paste made up of masoor dal or lentils or pulses. So probably this indicates that in ancient times, particularly in India, so whenever there was some affection by smallpox, the goddess Sitla Mata would have been regarded, venerated, worshipped, prayed upon, and she would provide some kind of a relief. It was being that way. And there has been some documentation from other Puranas also. In the Skanda Purana, in chapter 12, it has been quoted that for the sake of quelling boils and blisters of smallpox, and for the sake of the children, a devotee takes masoor lentils measures and grinds them, due to the power of Sitla Mata, children become free from the disease. So it's quite interesting. And this is something
Nawab
that,
sir, Nawab
yes,
sir,
yes.
your voice is not clear, sir. So the participants are posting in the chat box.
Is it okay now?
It is we are getting resound, sir.
Okay. Should I switch off and then join again? What do you suggest?
Yeah, yes, sir. That may be fine, sir.
Hear my voice now.
Yes, sir. It is okay, sir.
Okay. Can you see the presentation?
Yes, yes, sir.
Should I repeat what I spoke right now, or should I carry on?
Yeah, if you can go to the back side, sir, just to go back.
In the slide.
One
Okay.
slide back. Yes, sir. Please.
This is okay from here.
Yes, please.
Okay. So I was referring to the statue of the farm, or I mean, the mummy of farmer of Moses 5. Recently, scientists then found mummy, very old, 3,000-year-old farmer of Moses 5. We could detect that he also suffered from smallpox. If we can appreciate here from the picture. But he also suffered from smallpox, and smallpox has been responsible for causing diseases and maladies in humans from thousands of years back, and maybe some even civilizations beyond the first, that is the Egyptian civilization. And there is a possibility that these pox viruses have really affected humans. And the earliest record comes from China about smallpox from the 4th century after birth of Christ. And from India, we have documentation from the 7th century. And in the Asia Minor region, we have documentation about smallpox from 10th century and likewise. Now there has been a specific goddess. She is being regarded as a reincarnation of Parvati, and she is called the Sitla Mata. Sitla in Hindi meaning means to give some soothing sensation. In fact, during smallpox infection, there is a rash and there is a burning sensation on the skin, and it is believed that when someone prays or repents or visits Sita, she will be providing some cure to all types of poxes and sores and pustules and other diseases, particularly associated with smallpox or any types of dermatitis. Second, if you see her vehicle is a donkey, and she will be carrying a broom with Jharu on her right hand. And this Jharu or the broom signifies that probably she provides some kind of a soothing sensation, or probably she also, cleans off the dust and dirt from premises. And in the other hand, she will be carrying a pot of lentils or mussoorie. And it is believed that probably in the ancient times, in India particularly, people used to not only pray to Sitaram, but also apply a paste of lentil balls or masoor dal in order to give certain emollient properties and soothing effect on the skin. And then there have been also documentations in the program also. In the Skininfectionsinsheep chapter 12, there are mention about the effects of masoor dal and blessings of Siklamata. It's written this way: for the sake of quelling boils and blisters of smallpox, and for the sake of the children, a devotee takes musul lentils, measures and grinds them. Due to the power of Siklamata, children become free of the disease. So it becomes very interesting to know that how smallpox has, in fact, rearranged our even the pantheon of our gods and affected even the poor people and even the pharaohs, the kings and the peasants were equally affected. And not only in humans, pox virus has been responsible also for many epidemics in mammals and as well as in other mammals. Now, the picture on the left here, this is what represented smallpox, one of the most dreaded diseases and which had led to and mobility in those times. This here is a child from Bangladesh, and you can see the typical plaque lesions on the skin, and you can very well appreciate what was the type of suffering in case of people and animals alike. Now, this after that, there was Dr. Edward Jenner, who found that in certain unique ways that this compounds virus lesions from them while they could be regulated and incorporated in animals, they give them to cause protection against smallpox. And this developed the first vaccine, the smallpox vaccine, and which later on was more refined and sustained program. The smallpox eradication program was initiated. We could, smallpox globally, and we had really eradicated smallpox from the face of the earth, except for some select laboratories. This picture here shows a child, an 8-year-old child. Her name was Rahima Bruno. She was the last victim of smallpox, and when she recovered, there were no more reports of smallpox. Now, this is just a history of pox variety, and how we have shared humankind's sufferings, and also it reveals certain sufferings what the animals are experiencing now. Pox viruses are two basic morphology. One is the weak-shaped pox viruses, and they are largely pleomorphic, and they do not have a very definite shape, but they have got, they are very large, in fact. They are around 220 to 450 millimeter. Now, these dimensions are even larger than some of the viruses, in fact. So this is one virus, the pox virus, which size can even beat those smaller bacteria also. It has got certain surface tubules which are haphazardly arranged, and this is the electron microscopic picture here. And if you have a cross section, like sometimes in transmission electron microscopy, if you have a cross section, you'll find that it has got a core here, and this core represents the double-stranded DNA material, the nucleoprotein, and here we have a hollow. This is called the core membrane. Here we have a core membrane, and here we have got two lateral bodies here. And here we have and below. So this is a virus. In certain pox viruses, particularly in parapox virus, you'll find that they are ovoid and they hold their shape. They're not pleomorphic, but again they are the virus. To beat some of the smaller bacteria also. They are 250 to 300 nanometer in diameter. For certain, very beautifully arranged spiral filaments on their surface. Now, these spiral filaments give them a very, very characteristic shape. And this shape looks like a ball or it gives a very beautiful uniformity to the virus. Will not be spending much time on the pathogenesis because I have been forewarned about being too technical. But then again, I will just give you a rough idea about the most standard DNA that these post-viruses carry. Now, generally, viruses, they have a very small genome, particularly the retroviruses. Some of these viruses, they are not complete because they do not have all the genes that are required for replication of their genome. So sometimes they are regarded as incomplete viruses. But the pox viruses are some of the viruses that are complete viruses. They are not dependent in any way on the cell machinery. Usually what happens, a virus when it invades a cell, a host cell, it sometimes sabotages the machinery of the cell and uses some of the products of the cell itself or shuts down the factory of the cell itself. And we use their genes and proteins and other enzymes for their own benefit. But here in case of the pox viruses, they are self-sufficient in the sense that they carry everything that they need, everything, starting from the early genes, the intermediate genes, and even some of the late genes also. So they have everything completely. They're not dependent upon the host cell. In fact, they have got, this will be discussed later on, they have got certain machinery that they can also sabotage or, system, block them, invade their immune system, and will be self-sufficient to replicate itself. And there are different ways by which we'll be discussing this again sometime later. So then how these pox viruses or these envelope viruses, they attach to the cell surface, how they get rid of their envelope, how the genetic material is transferred into the cell, and how these first lipoprotein they follow, and how they synthesize their first the proteins, the early proteins, the intermediate proteins, and the late proteins. So these are sequential events that ultimately lead to the production of the small baby viruses. We call them as the virions. So these virions, one type of virion we call is as the mature virion. And this mature virion is the infected particle. Now, this has already matured and is capable of causing further infection of other cells. Okay. Now, in that, once these viruses reach their mature stage, they might, acquire certain more atomages over themselves, or coverings over themselves, or a cloak. These cloaks or other materials, they usually use from the body material or even the endoplasmic reticulum, and we call them as the intracellular envelopiruses. So here you can see that this is an intracellular envelovirus because they have acquired certain of the cell, the membrane from the Golgi apparatus. Sometimes they are associated with the cell surface. We call as them as the cell-associated envelopiruses. Now, sometimes they will just go into the cell and acquire a part or a layer from the cell membrane. We call it as the extracellular envelopiruses. So these are certain intricacies that we should not be too much concerned right now. But again, they are of interest and they are of importance during viral pathogenesis. Now, we will be moving from the general discussion of pox viruses and we'll be moving specifically to the pox viruses. Now, the general pox virus causes commonly a disease which is Again, known by many names, it causes contagious eczema, or also what we call as pons. It comes, the word pons comes from the Nordic word krufa. So that krufa means cattle, or even sometimes it means scabs. It is also sometimes referred to as sorelock, particularly in New Zealand, and pustular dermatitis or contagious pustular dermatitis, or even sometimes referred to simply as scabby mouth. So there are different, local names for disease. I won't be knowing the particular names in India, what people in Telangana or people in Jammu Kashmir or people from other states they usually call, but in your language, from Assam, they sometimes have a very non-specific name, which is sometimes confusing again with affinity. So these viruses, usually affect the ungulates and other domesticated livestock. Now, see, this is a chart that represents the different species of the Monkeypox viruses. The first one is a bovine papulosis communities virus. So this here represents the BPSV. Okay, this one, this picture. So it causes a vesicular type of a disease, and sometimes we might sometimes confuse it with some other diseases like FMD also. But again, FMD is a vesicular disease, but this does not form vesicles clearly. We have the olive virus, which we will be discussing more now. Then we have a parapox virus of the reindeer in New Zealand. So this here is a parapox virus of New Zealand. This has been considered as a different species with certain variation from the normal or virus. Then we have a pseudocarpox virus, or sometimes we call it as a mimical snowmobile virus. Now this also is a zoonotic virus. So this is mimical snowmobile virus. Then we have certain other species. Last time 2018-19, they were included as distinct species within the parapox virus genus. But again, recently, 2021, I checked ICTV online and we found they have been, only temporarily restricted and yet verification about them being specific or different, species as yet is yet to be confirmed. So when we have the squirrel parapox virus here, then we have the camel contagious external virus here. Then we have Chemois contagious external virus. If you do not know what Chemois is, it's a type of a wild goat. This one. Then we have silk virus or silkpox virus. This usually affects the mammals, the wedding cells, etc. So these are all caused by the parapox virus genus. Now, these viruses, specific viruses, are considered to be, or will be given the status of a species, some of them. Of course, this nomenclature keeps on changing, like I told you, from year to year, depending upon your deliberations by ICTV every year. This is the genetic map of all virus. So I do not want to go into the intricacies, but I'll just give you a fair idea. So if supposing this is the entire genome length, so this core region, it represents a very unique conserved region because here, many of these parapox species, they will share, will be homologous there. They'll be sharing these nucleus, the nucleoside. And then only at the terminal portions here, of course, we have the ITMs at the both ends. So this region here, usually they are less conserved, and these represents certain variable genes, which are generally associated with specific pathogenicity of the viruses, how they infect the host, how they enter the cell, what are the virulence properties, what are the host immune evasion properties, etc. etc. If you see here, because we'll be considering the economic significance of the virus, of the virus. in contagious eczema does not cause a very large scale economic, setback. But again, if the suffering is very acute or the infection is very acute, for example, the picture I'm showing here, this is the picture of a sheep, and you can well appreciate the appalling situation of the animal here. If in such a situation, the animals, they cannot eat, they cannot breathe, there will be quite so much of suffering to the animals, and also this will lead to misery of the organisms. Yes, the production potential of this animal, they also goes up. Okay. This is particularly true. There is a considerable economic setback, particularly in those farmers that have got a very small stock population, depending on their livestock, and their livestock is very severely affected with just a climate or wolf. And this is also seen in certain arid regions where goat husbandry or sheep husbandry becomes a very prime source of economy. And also in certain situations, because these are considered as small and scale. So in certain situations, when the farmer is very poor, and his farm stock is affected with such a severe type of infection. So in that case, where the input is very much less or zero input economy farming is there, so it will definitely lead to very severe economic crash to the farm. So mortality generally is now 1%. So this is not a rare disease, so to say, and it is not noticeable also according to OID. And then again, if sometimes the infection is very severe, depends upon the virulence of the virus, and the second thing is about the immune status, or let us say the prevailing managemental policies. And the morbidity is very high. Now, why the morbidity is high? Because these animals, they are always kept in a group, and they always lose each other. And even in case of the young ones, when there is affection of the pigs, they will be suckling from their milk or their mother. And in such cases, the animals or the young ones, particularly the suckling ones, become extremely susceptible to infection. And in case of the young ones, you will always find that the infection is very severe. Seriousness of the disease is also sometimes, not much of interest. Why? Because this disease is spontaneously regressive. It regresses by, say, around 30 days, or say sometimes even only as 3 to 6 weeks. So if there is absence of any complications or secondary infection, usually what happens, they heal spontaneously. And animals that are healed, usually they are resistant to a secondary infection, so within, say, 3 months or up to, say, 6 months. And then again, they are also susceptible to reinfection, because like in smallpox viruses, say for example, in case of, say, smallpox, or probably in case of goatpox or sheeppox, animals that are once affected generally do not get affected anymore. This is a general thumb rule. But in case of smallpox virus or in ours, you'll find very strong, because I will be discussing it later on. Immunity in case of smallpox virus is generally cell-mediated immunity. So that is the main type of immune response that generates in the face of an in face of an infection. So the neutralizing antibodies are very less. Now, in case of absence of complications, and say bacterial infection, or let us say in case of infestation and maggot formation, or if there is no such complications such as mastitis, so the animal may not be very severely affected. Production losses may not be much. One thing we should always bear in mind: this disease has got a zoonotic significance. It can spread to humans also, and therefore it is considered as an occupational hazard. The disease epidemiology, the scabs that the animals shed during healing, once the animal is affected, they usually, progress into formation of scabs. And these scabs are the storehouse of the viruses, or these are the viral factories. So once these scabs they fall onto the ground, they can survive for months and sometimes even years. And in cold places, it can winter also. When it overwinters, it can subsequently affect the same flock of animals because the immunity is not very strong. Neutralizing antibodies are not very strong. So they can keep on infecting and reinfecting the same animals or the same flock of animals or the same area, and it leads to generally endemicity. Immunity is very short-lived, I already told you. And fourth is sometimes uh, considered as a goat disease rather than a sheep disease. Not that sheep are resistant, they are all equally susceptible, but generally there is a predilection for affection of goat case of sheep. And like I told you, the young ones, the kids particularly, or the lambs, let us say, they are very highly susceptible. Now, sometimes there are certain predisposing factors. For example, if the animals are under stress or if there is any immunosuppressive condition like prolonged therapy of corticosteroids or there's a secondary bacterial infections or even in case of other nasal infections like your PPI. And sometimes even if there is some trauma like infighting, ear tagging, or castration or perivoting. So whatever brings a compromise in the continuity of the skin. And if this virus is endemic in that area, any broken skin might lead to, for this of infection like off. And in case of humans, those farmers, those veterinarians, I will be showing my slides also, are affected by all virus. Adaptive slot people that are associated intimately with sheep and goat, including butchers. So there is a chance that if there is any broken skin or there is any break in the integrity of our skin, we might also contract the disease. So in case of humans, you'll find that there is a solitary non-healing type of lesion very on the face or on the axilla. So these here represents some of the other species which can be affected by, potentially affected by, transmitted from sheep and goat to other animals also. So there are instances where the domestic cat, dogs, sailors, even in case of India, we have reports from Mizoram and in reindeer, that is the reindeer, and in muskox in America, and the kafin, this is in China, and the mountain goats elsewhere, bighorn sheep, cow sheep. So these are actually wild sheep. So they can, or they have the reports that these animals or these species of animals have contracted the of disease from diseased sheep and goat, domestic sheep and goat. So these are the reasons generally. So here you see, this is a pathology. I'll be discussing about the pathogenesis of the disease because pathology means pathogenesis. It starts with pathogenesis, it establishes as a definite pathology. So this is a petunia, this one. And these are scabs. Of course, these are the intermediate stages, and this is a mature scab. And this mature scab is a viral factory. Sorry, I did not mean to say a viral factory. It is a storehouse, a ground for the virus propagation. These are again viruses. Now these the virus, the scabs are shared here in this area, and this is the underlying healing scheme. And sometimes when the scabs fall out, it might leave a raw bleeding surface also, particularly when the animals, traumatize themselves. So this is a healed scab. These are all healed. And this is scab lesions are also present. Pimple-like lesions are also present on the other teeth. Now, this is important because dams that are usually suckling, milking dams, they might feed their young ones, and these young ones, 100%, the disease will get contracted to them. And this here today, Dr. Nawab, Dr. Ashok, he showed me a picture of a kid affected very severely. His buckling mucosa and tongue was affected. And he was asking me to confirm whether this is yes or not. So this picture here, these are some of the very overt complications of PONF. This is very, very severely affected. And the kid here that I am showing, it died after 2 days because we tried our best. We tried to feed him, bottle feed him, but ultimately we could not uh, uh, save him. Then this is one type of very unusual affection. Here, on the ear, we found that certain, alopecia was there. We primarily thought this was a mycotic type of infection or probably scab, mite infection was there. But later on, when we did PCR, we found out that these scabs were nothing but those of parabolic. Here, the geometric lesion we can see here. So these are generally present on any compromised skin in case of humans. So this was the picture of a friend of mine who was working there, and he was helping me out. And he contacted this orb, and later on it developed into a pustule. And we took some samples and we did PCR and we confirmed. And this is me here. I also contracted certain, of these lesions while working with off. Yes. Off is an epitheliotropic virus, in the sense that it only affects the keratinocytes or the skin epidermal cells. In vivo, or inside the animal or inside the live animal, there is no tissue other than the keratinocytes that can support the growth of the layers. But under in vitro conditions, we have many types of many cell types, including lung testis cells. Then we have kidney cells, primary kidney cells, and sometimes cell lines also. Even in case of donor cells also, sometimes some people they have successfully grown virus in these types of cells. But in vivo, this is very unusual for this, or very typical, let us say, or unique of this virus, that it only grows in the keratinocytes. And usually the sequential progression of the lesions is through the formation of an erythematous macule. Then it progresses into a papule. Then it forms a vesicle, then a pustule, and then ultimately, when it heals, it forms a scab. Now, these pictures here I have shown you, this does not represent us, but I am just giving you the idea what erythematous macule looks like. So, a macule is a local area of discoloration. So, it sometimes may be reddish. When I say erythematous, it becomes reddish, or sometimes it may be blackish. So, it starts this way. Then it forms a papule. Papule is a very, very small type of nodule. Then it forms a vesicle. Okay, so vesicle formation generally suggests that there is hydropic degeneration in the keratinocytes. Then, after some time, a vesicle will form into a pustule. Now, pustule always signifies that there is secondary bacterial infection. And after it heals, it forms a scab. System lesions that are associated with all virus infection is acanthosis. When I say acanthosis, it means that there is hyperplasia or irregular thinning or thickening of the epidermis. Then there is hydropic degeneration of the spinosome cells. You already know the spinosome cells or the pickle cell there. They are star-shaped, and they have got certain interdigitations to which each of the cells they hold fast to each other, And when there is hydrotic degeneration or accumulation of water within these cells because of a failure of the sodium potassium pump, so these cells they will swell up. Once they swell up, they lose their interdigitations or cell-to-cell contact or attachments, and these cells become individualized. And one time, some of the cells will rupture also because of excessive edema or cellular edema, and then it forms a vesicle. Then there is also hyperplasia of the basal cells. Now, generally, why a scab forms? A scab forms in chemotherapy. Now, once the body comes to know that the epidermis has been attacked by an infectious agent, for example, the pox virus or the off virus. So once the epidermis are affected and these cells are taken over by the virus for their own replication and multiplication, the underlying dermis comes to know because there are certain signals that are sent by the epidermis or epidermal cells. So the dermis, what it does, it primarily tries to block the blood supply to the above epidermis. So this is when a local ischemia results, or ischemia is a loss of blood supply. So the thrombosis of the blood vessels in the dermis, they lose or they cut off the blood supply to the above epidermal cells, and these epidermal cells they die. This is the ultimate sacrifice that the body does or gives to shed off the invading pathogen. So once there is ischemia, there is loss of blood supply to the overlying tissue. The overlying tissue it dies, and it forms a scab. And this scab will at one time shed off fall off. There may be some bacterial infection also, because it forms a very good medium for bacterial growth. And ultimately, a new epidermis will grow from beneath. So this is a part of our own immune, cell-mediated immune response. So this is why in most of the pox viruses or pox virus infection, we find formation of a scab. This is one thing that we have to remember in case of parapox virus only. Pathology is restricted to the skin and no systemic spread is there. Unlike your capripox viruses, for example, your goatpox virus and even your LSD and your sheeppox virus, because they have got intermediate stage, where the virus may enter into the blood circulation and attain systemic infection. This is something that I think we should probably skip. Probably many of you might not comprehend. Of course, we can just have a glance. This is the bacteria, the virus. It will attach, it will enter, it will uncoat itself, then there will be replication. And remember, the inclusions of pox virus, any pox virus, will always be intracellular. This is the nucleus here. So the nucleus will not be invaded by the pox virus because the pox virus always carries everything it requires. If it has to make DNA, it will have its own shower, it will have its own masala, it will have its own meat, everything it carries by itself. It will not be dependent on the cellular machinery. In fact, it has got enough proteins and enzymes and other genes to sabotage or even shut down the normal host cell. So that will never go into the nucleus. So pox viruses always you'll find the inclusion. What is an inclusion? It is just the virus factory. Factory or the viroplasm, we will always find in the cytoplasm. Then in the cytoplasm, the mature viruses form or the virions form, these viruses. Now these viruses, the mature virus, is the infective virus. And sometimes what happens, it might acquire an additional protein from the Golgi apparatus. So this is the Golgi apparatus, or sometimes from endoplasmic reticulum, it will wrap some of its proteins over itself. This is what we call as a rat virus. And sometimes this rat virus may be associated with the cell surface here. We call them as the cell envelope virus or cellular envelope virus. Sometimes it will help it will use some of the acting cytoskeletal machinery to use the cellular cytoskeletal machinery, the acting filaments to climb out of the cell. And once it does that, it will also require some of the additional cell membrane over it. Now let's see what is the host response. How does the body react to invasion by the virus? So the first line of cellular defense, as we all know, is the neutrophil. So the first time that the virus invades into our body, there is this very strong chemolytic response by the neutrophils. Then there will be accumulation of the dendritic cells. For uninitiated, dendritic cells means those antigen-presenting cells. Then we have the lymphocytes, the T lymphocytes. There are different groups and subgroups of the lymphocytes, T lymphocytes. A particular subset we call as the CD4 cells or helper T cells. Then we have the cytotoxic T cells, the CD8 plus cells. Then we have the B cells. The CD4 cells and their products, particularly interferon gamma, that is usually exuded by the CD8 plus cells, they help in protecting against infection. So the interferon gamma is a very potent weapon in which these cells try to kill the bacteria, and if they, the virus, and if they cannot kill the virus, they will at least kill those cells that have been invaded by the virus. Okay, so the CD8 cells, by virtue of recognition for the MSC class 1 molecule, so they will try to kill those infected cells. Then the response of the B cells is extremely limited, very, very limited. And therefore, we do not see a neutralizing antibody response, not a very big neutralizing antibody. And even if antibody form one time, and the peak is not much. And the neutralizing antibody that is usually synthesized by an infected animal or a convalescent animal, this is sometimes considered to be not sufficient enough to impair immunity to young ones. So either through colostrum or through passive serum transfer to other infected cell animals, they cannot be considered to be very effective. Let us see. We have already discussed here the cellular response, the host response. Now let's see, because these viruses are very funny. One of the most cunning viruses are the pox viruses. So no matter how much the body or the host tries to contain infection, there are so many tricks and counter tricks by these viruses that can help to evade the immune system. So these, a long list of, there are even more, there are many more, viral proteins that are synthesized. For example, here, VEGF, this is a viral endothelial growth factor. So what it does basically, even if the body tries to cut off its blood supply, so the virus, it knows that the body wants to cut it off. It wants to form the skin. So what it does, it synthesizes its own type of endothelial growth factor, and it helps the cell, which is under its domain now. So the cell will, through by virtue of the virus, it will start synthesizing the VEGF. And what this VEGF will do, it will go and signal the nearby endothelial cells and ask them not to stop their functioning. Then we have different type of anti-inflammatory or pro-inflammatory responses. Even there are certain proteins that are synthesized by the virus that will cause apoptosis of the B cells also. For example, here. So even if the B cells come and try to form antibodies against the virus, so the virus affected cell, by virtue of the virus itself, it will synthesize certain proteins that will kill the B cells. Or sometimes they will also not allow recognition of the virus-infected cells by the cytotoxic cells. So these are some of the counter mechanisms by the viruses itself with immune response. Now remember one thing, in case of parapox or orchid infection, immunity is always or primarily cell mediated. This is something that we should always bear in mind. And that is why immunity is not strong, vaccination is not successful. Now, in histopathology, this is the picture of a scab. And inside the scab, you'll find, different progressive lesions. And one thing we will always find, of course, it depends upon the stage of infection, intracytoplasmic eosinophilic inclusions in the epidermal cells. तो वहां पे जितने भी epidermal cells है, इसके अंदर हमें दिखेगा these intracytoplasmic, very large intracytoplasmic eosinophilic inclusions. In fact, some of the largest inclusions caused by the viruses are from Avipox virus, what we call as Bollinger bodies and Borrel bodies. So Borrel body is the counterpart, avian pox virus counterpart. This is the entire inclusion, what you call as the Bollinger body. And it is made up of Borrel bodies. So Borrel bodies are nothing but the viruses. But please remember, the pox viruses are the only viruses that we can see under the light microscope. They are so large, even larger than some of the bacteria there. We can see them under the microscope. So this picture here at D, it represents, of course, there are three pictures. We have tried to, make them a montage here. So this is a composite picture. It shows many different lesions together. So this here is a vesicle, if you can appreciate. This here is a vesicle. This is the epidermis. This is a vesicle. This also here is a vesicle, fluid-filled vesicle. And this is the scab. And beneath here, the epidermis is already regenerating. Here. Now come to diagnosis. Serological diagnosis, it is not successful. If you say EGPT or precipitation test, no, it is not successful. We have tried many times, and eventually one day we could, through hyperimmune sera and antigen filled sample or clinical sample from the field. We had raised the hyperimmune sera. We tried to go for normal EGPT, but there was no reaction whatsoever. How many times we tried, no matter. Then ultimately we went for counter immunoelectrophoresis. We had applied 2 electric field goat hands, and only on one time we could be successful to precipitate this antigen antibody complex. So serology is not a very good idea about diagnosing field diagnosis of or bedside diagnosis or clinical diagnosis of all virus. But we can definitely go for ELISA also, which I'm not showing here. And immunochemical, always there, immunofluorescence. Yes, if you see, these are the keratinocytes, and this dark area here, it represents the nucleus, and you'll find that the large accumulation of the virus you'll find in the perinuclear region inside the nucleus. Here again, peroxidase technique. This is a very good technique. Of course, the efficacy of hyperimmune sera would be a little less. We were using mostly monoclonal antibody, which we had commercially acquired. And cell culture, I already told you, in vivo only supported, virus replication only supported by the keratinocytes. But in vitro, we can have a large selection of cells which can support the growth of parapox viruses. This is not only in case of virus, even the other parapox viruses we can successfully grow here. So here we were using two types of cell. One is the primary kidney cell and primary testis cell. We were using a specific lamb testis, embryonic lamb testis cell acquired from APCC. This was a very good cell that supports the growth of this parapox virus. And this is the cytopathic effect. You find that there is rounding and there is rounding and very typical CPE you can see. The same cells, when we stain, we can demonstrate very beautiful inclusion bodies. So these are all inclusion bodies here. So this is the cell here. This is the nucleus. Just outside the nucleus in the perinuclear region, you'll find inclusion bodies. So these are the first inclusion bodies, and subsequently upon second passage or third passage, we could grow very, very large inclusions. And immunofluorescence is there, same cell lines. We have the immunofluorescence test. Then for PCR detection is very easy. Mostly the B2L gene is used. So this is the B2L gene. So what is the B2L gene? It actually, that gene encodes for a particular mesoid envelope protein. This is called as P42K, and this is homologous to our vaccinia virus. And homologous protein is called as P37K. So this gene is widely used for detection of parapox virus and parapox virus. It will diagnose for your organisms. For example, your reindeer virus and the seal virus. All species from the parapox virus genus can be detected by this system. So it was developed by many different workers. Inosama is one, then Hosamani is one. So then again, the other gene that is usually used is the GRF gene. This is another target where we can design PCR primers. And also we have another gene as called as the A32L or the ATP gene. So these genes are usually used for detection, confirmation, as well as for genetic analysis or relationship between different strains of the virus. We tried our hand on this electron microscopy. This is a scan, and under the electron microscope, we can see this is a viroplasm. If you can appreciate here, this is a viroplasm. So this is another. This is a composite picture. Of course, there are two photographs. We try to join them together. And this here represents the epidermis. This is a scan region. This is the epidermis. And within the epidermis, we can see the viruses. Here, if you can see the dots here, these are the viruses. And again, we had many of these viruses, we try to measure them, and you can see, really appreciate, this is the main virus, this is the envelope. And in certain clear sections, you can appreciate the core, viral core, as well as the lateral bodies. And you can appreciate the size of this virus, 263 nanometers. This is quite large for a virus. So these are all viruses. So these are associated within the cell, and this is outside the cell. And I have tried to mark the immature virions and the mature virions and the cis. So this is a virus. So this is the virus core. This is the cloak or the additional membranes that it had acquired from the Golgi apparatus and the area. This is another virus, the viroplasm or the viral factory. This here represents an extracellular envelope virus. So this is the cell here. So this virus has already escaped from the cell, and it will be present in the scab, or it will go out in the environment. And this here represents a virus that has used the actin cytoskeletory network of the cell to strand out of the cell. That was all about transmission electron microscopy. Now, of course, it is very costly, takes time. It will take, say, around, depending upon how far you are situated from your electron microscopy facility, it's costly also. And time consuming. But then again, this method, this is what we call as a negative staining electron microscopy. This is very, very easy. You can directly collect sample and just submit to the electron microscopy lab, or you can process it on your own also. So all you have to do is, use some very heavy elements for negative contrast. For example, you can use phototungstic acid or molybdenum. So what it will do, it will negatively give contrast, and you can appreciate here the beautiful crisscross hatching or the fibril outside the. And there are actually two forms of the virus. One is called a C form. The C form, you do not visualize the crisscross fibrillar pattern. Otherwise, most of the virus. This is another type of microscopy, what we call as the immunoelectron microscopy. So usually the viruses are tagged by an antibody, which has already been conjugated with immunogold particles or colloidal gold. These are the viruses we have successfully tagged. This is also very easy, but then again, you have to use the tissue samples. Again, tissue samples, it will take time. And any electron microscopy procedure, it involves a significant amount of cost. But again, negative within an hour also, you can give your confirmatory diagnosis. Cryo section. From cryo sections, you can keep, say, within minutes, you can give. For example, if you have a skin scan or any tissue, you can make your cryo sections and immediately for any stain you can use. Even normal hematoxylin eosin you can use, and you can demonstrate the inclusions. Here we were using FITC, and this is IPT. These so far we have discussed about the field clinical material that we have collected. So what we tried next time was try to establish this disease, the same disease in live experimental animals. So we took some goats and we simply abraded the skin or injured the skin with sandpaper, and then we applied a solution of the clinical sample over it. And right from the second day onwards, we were seeing that the lesions were starting to develop. And by the end of the 20th day, we saw that complete healing was there. So 28 days is 4 weeks. So within 4 weeks, we could see, these are the photographs here. We gave some abrasion here, or sometimes even, a small scar. And we simply applied the clinical material, virus material. And these are the progressions. See, in the second day, third day, fourth day, fifth day, sixth day, and then 8th day. By 20th day, lesions were very, very prominent, but after 28 days, complete healing. So these are the progression of lesions. So initially, on the first day, or say the third day, we can visualize the inclusion body here, and hydropic degeneration we can see. And third day, again, many of the cells, these are the keratinocytes. So these are the basal cells, these are the keratinocytes, and you can appreciate that there is considerable hydropic degeneration. And some of these cells, they have already ruptured and will form vesicles. And also there will be secondary infection. So these are bacteria. So these are secondary infection by 4th day. And 3rd day onwards, you'll find that the inclusion body starts. But around 7 to 10 days, you'll find that the inclusions are absent. So if you collect clinical material and submit for histopathology, if the lesions are old, say beyond 7 days, you may not find the inclusions. And if your diagnosis is based on finding inclusions and you do not find them, you might have a false negative confirmation. So here again, see by fifth day, so many cells they have undergone hydroponic treatments. And also here you see that by fifth day, in some other sections, you find that there is considerable, ischemia or ischemic necrosis, as well as infiltration by large number of cells, infiltrating cells, inflammatory cells. So here you see, so this is the entire scab area. This entire scab area will fall off because here, somewhere around here, all the blood vessels have been blocked by the body. And this is the amount of infiltration that we generally see. See here that we are seeing, because this is an 8 day, so these are all macrophages, all macrophages. So you see here, vesicles, 8 day, macrophages here, and this is almost healing in some areas. And by the end of 20th day, you find that the entire skin is normal. Try to reproduce this disease in some experimental animals also, laboratory animals, or in case of, say, mouse, in rabbits. And some of the workers, they have been successful in establishing the disease in other animals, in cats, in dogs, in mice, in rabbits. We were not successful, but at least we got a very, very, and so these are some of the scarified skin tissue. If you see a rabbit here, we had put some scarifications here, and then we applied the clinical material, but no disease. Similarly, here in case of mouse, we tried some scarification, first 0 day, first day, second day, third day, and it healed by 4th day. We could not establish. But very small pinpoint punctate vesicle we could find. Punctate meaning very, very small vesicle we could find. Third day, by 4th day, it replaced. So treatment of so therapeutic, we can use topical, any type of topical disinfectant, parenteral antibiotics, just to check secondary infection, because we cannot treat viral disease with antibiotics. Supportive therapy is very important, and managemental intervention is very important. For example, particularly in the young ones, where they cannot drink or where they cannot eat. So probably they need some intervention, some. Then differential diagnosis, this is extremely important. There are certain conditions with which we should try to differentiate orophytes. One is ulcerative dermatosis. Of course, this is a bacterial predilection. Then mycotic, or unusual, if not common. Then we have eczema and foot and mouth disease. Remember, we should see for or check for vesicles. Then papillomatosis, this is important, or any other type of proliferative manifestation. Sheeppox is important. Goatpox, both sheeppox and goatpox, and Bluetown is important. Then prevention, biosecurity. Of course, if you have in a farm already there is infection is there from previous year, control is very difficult. It should be very frankly, I should speak, because the farm premises are filled with scabs, and these viruses, enveloviruses are there, and they can survive for a pretty long time. So if you have to ensure that the farm premises are to be disinfected, so we should use proper managemental interventions. Colostrum does not help you, does not help secure immunity in the lambs, or may not. Vaccination is doubtful. Of course, there are some vaccines available in other countries, particularly in in New Zealand, in the certain European regions, and also in Africa. I have seen that they have been using some vaccines, but I do not think in India we have a vaccine for off virus. And we should not be using a vaccine because primarily I have been telling off and on the immunity or immune status is basically cell mediated. Humoral immunity is very, very low. And there is no successful neutralizing antibodies. And animals are prone to infection and reinfection. So if you can survive or if you can consider or to be limited disease, or probably if the lesions are not very extensive, if the mortality rate is very, very less, probably we have to leave it off. Then again, sometimes there may be certain complications. And in that case, we need, supportive therapy. We need better management. Next topic is Capripox virus. So because my primary talk today was on off virus, and already some earlier speaker had discussed about Capripox virus, about goatpox and sheeppox. What I will do is share my experience, what we did in Jammu. So this is not my primary topic, just because I have to discuss the pox viruses that affect sheep and goat. So the disease that we of our interest was today all virus. And next is goatpox and sheeppox, and also about LSD, which of course is not a sheep and goat disease, but then again I might be or I will try to answer if you have any queries regarding LSD. Of course, we do not have much experience on LSD because this is a new disease, as recently in India. Our experiences with Capripox virus. So this is also pox virus and infection with Capripox viruses, either LSD or sheeppox, sheeppox or goatpox. It is a notifiable disease according to OI. So whenever you have any reports of goatpox, sheeppox, or LSD, immediately you should alert the authorities. Transmission, direct contact. In case of sheep and goat, we saw these clinical signs, and it is very easy to say diagnose for all of because the reasons are self-explanatory and very, very conservative in a way. But again, sometimes we might be confused with certain other diseases which we have already mentioned, and we should take, sometimes it needs certain laboratory, maybe even the suggestion or consultation of an expert. But in case of sheep and goat pox, sometimes it really leads us into circle, and unless we have a very firm laboratory confirmation, it becomes sometimes difficult to diagnose sheep and goat pox. Clinically, you find conjunctivitis, fever, rhinitis, anorexia, depression, lowering of production, and ocular discharge, and blepharitis, and lipopurulent nasal discharge, and dyspnea, and labor bleeding. So all these things you will find in sheeppox and goatpox. Because remember one thing, all these Capripox viruses not only affects the skin, it also has systemic predilection, also can disseminate into most of the visceral organs also, unlike your off virus. So this will skip. Clinically, you'll find that there is blepharitis, you see here, then lipopurulent nasal discharge you find in both sheep and goat, and same progression of lesion. First you find a methule, then it forms a papule, then it forms a nodule, and sometimes a vesicle. But vesicle stage is generally more reserved for vesicular diseases like your FMD. Even if you can appreciate vesicle, this is only under a microscope. Then ultimately it will heal and form a scab. These are pictures from the sheep. These are pictures from the goat. This is a papule. This is a nodule. These are some of the scabs, and these are the raw pock lesions once the scabs fall down. So this stage here depicts a convalescent state, and it represents considerable, um, uh, healing. Teeth, luminal lesion, perianal lesion, tail, these typical pock lesions can be found. Case of the respiratory system, same, we find large nodules, pneumonia, and irritation, and then some raised areas, whitish, grayish areas you find. And these are some of the representative pictures in goat also. Then in the tongue, you can see here the very definitive pock lesions and grayish white nodular lesions on the liver. And even in the abomasum, I don't think the picture is very clear here. Otherwise, this was a very nice lesion. Beautiful, pock lesions we could find on the abomasum. And sometimes it may be associated with some form of enteritis also, may or may not. Uh, Same, this white grayish nodular lesions you find on the liver. And this here is a very beautiful picture here in the abomasum. You can see these white grayish circumscribed. Kidneys also same white necrotic foci, grayish, or sometimes even frank nodular spots you could find in the cortico-medullary junction. I do not think you can appreciate very much here. So you find here small, necrotic foci. I do not think you can appreciate. We tried to grow them. We did not have cell culture at that time. So on an emergency basis, what we acquired was embryonated eggs from chicken, and we gave passas. And for sheeppox virus, we could not establish infection, but with goat pox, we could establish by third passa. We could see beautiful. These are the histopathological lesions. Very big inclusions you'll find. Same as in case of parapox virus, you can observe the intracytoplasmic inclusions and hydropig degeneration and vesicle formation and acanthosis and lesions, as is typical of many of the pox viruses. Skin lesions. So here this represents this is a composite picture. There are 3 frames here. So you can see this is a pock lesion. This U-shaped area is a pock lesion. So we tried to include that area, and you see that there is total sloughing or loss of this epidermis here. Typical inclusions you can find. This is in the lungs. There is considerable consolidation. And one thing is that the normal pneumocyte type 1 is replaced by more hardy type 2 pneumocyte. You can see here, type 2 pneumocyte. So this is called as epithelialization, or sometimes even referred to as fetalization in pathology. Inclusions we can find even in the bronchial epithelium, just as in the skin. We can see these lesions in the lungs also, in the trachea, and even in the intestine. We see that there are inclusions, and there is a typical type of cell. These are actually macrophages that have got these very vesicular type of nucleus, very large, and these are called as sheeppox cells. So these here represent sheeppox cells. So very unique and characteristic of sheeppox or goat pox for that matter. This is in the abomasum. In the abomasum, similarly, you can see here we do not find much lesions in the epithelial surface, except for presence of certain pox cells, and then also plenty of macrophages. But then again, something very unique we found in the underlying blood vessels. in the arteries, we find that the endothelial cells are vacuolated, and sometimes we also find, something like inclusion which we do not confirm. And in the liver, so these were the circumscribed areas, white necrotic foci. So this here you can see this, and non-specific histological lesions in the liver. There was fatty change. There was vacuolation of the hepatocytes. Then again, something very, very interesting. In all the biliary cells, the bile cells, we could demonstrate these typical inclusion, large inclusion, all these large inclusion. And here we can see these are the toxins. All the biliary epithelium you can find is error represent. Of course, I do not think you can appreciate on your mobile phones. So inclusions were there. Same on the chorioallantoic membrane in the chicken embryonic eggs, we could find these inclusions. And we did PCR. In the PCR, we targeted a specific gene called as the P32 gene, which is very commonly used for confirmation, diagnosis, as well as for phylogenetic analysis of different Capripox viruses. Of course, this P32 gene diagnosis for both sheeppox as well as goatpox virus, and it should also probably, I do not know for sure, probably it will also it will provide for LSD diagnosis also. Do not take my word for it. We haven't checked it. And some phylogenetic analysis we tried. So this is one of our pox strain. This is the goat pox strain, and we could find that our goat pox clusters with the normal cluster or clades, and sheep pox clustered with the sheep clad. Now there are certain references. All references from Jammu and Kashmir, IVRI. They said that goat pox or goat can contract sheep pox or vice versa. Goats can or sheep can contract goat pox. But in certain literature, this is contraindicated. And it says that no, sheep can be only affected with your sheep pox virus, and goat can only be affected by goat pox virus, and likewise. Documentation. Yes, we have isolated goat pox virus from goat. We do not know, but at least the preliminary report from Jammu and Kashmir said no, there is a prevalence of sheep pox here, and which affects both sheep, because we generally see that the goat and sheep are always reared together, particularly here in Jammu and Kashmir, or in any state or any enterprise where there is or any we'll always find goats mixed with sheep, and you know the reason why. But we could find that our goat pox was different strain, and our sheep pox was a different strain. But very interestingly, we saw that the sheep pox that that is prevalent here was very, very similar to the vaccine strain or the parapet strain. And this is our goat pox virus. So you find that they are very similar to the vaccine strain, and also it is similar to the other out in the house. But our goat or subtle differences with our sheep pox, and also I think was more inclined towards those strains that are prevalent in some other Asian countries like in China. Now, why discussing pox virus is important? Why is it important? Pox viruses have got a very significant historical interest, and it is also significant in terms of public health. Because this is one virus, for example, your smallpox virus, your parapox virus, it primarily causes disease in humans or is zoonotic for humans. And it's been also responsible for extensive study of viruses and also led shaping our modern medicine. It also allowed the development of the first vaccine. And course, these viruses are very, very large and has got a very large, engineering or genetic engineering allows the formation of some recombinant vaccine, which is very large. Right? So some of the non-essential nucleotides, they can, some of the, interested very large viruses. So they can act as vehicles. And sometimes they can be also used, which has been used even now, of course, on a trial basis. Pox viruses have also been used in cancer therapy also. So those cells that are affected with cancer, so they are generally targeted by these pox viruses. Of course, these are genetically engineered pox viruses. What is the way forward now? So domestication of animal, we know, started very early. There are evidence, archaeological evidence that around 10,000 to 12,000 years back, domestication started, and humans, they started settling down. Instead of becoming a wanderer or a gatherer, it started, depending on agriculture and domestication of animals. Now, with domestication of animals, and when they started their coexistence with different viruses and new pathogens started evolving. Okay, and then it led to some of the very first endemics also, and some real devastating consequences. One of them being pox virus, which has been plaguing even the pharaohs of old, and sometimes it has also led to much belief, because this was the only resort that the poor people could afford at that time or in those ancient times. They were also rearranging their paintings of the gods also. And ultimately, it led to the development of the vaccinia virus, which also the variola virus killed so many people. Vaccinia virus, on the other hand, nobody knows of its origin. Kahan se aaya? Where vaccinia virus, what it did, it formed, or it was regarded as the first savior for humankind. And because the variola vaccinia virus, they have been so extensively studied, and it had opened the gateway for new insights in viral pathogenesis as well as host resistance. Now, parapox virus is not a very serious disease, at least I consider that, and it's very strong, and vaccination is not, successful, at least. But pox viruses are more severe and notorious, and it requires a little close attention. The members of these pox viruses, like a goatpox, sheeppox, and the emerging lumpy skin disease virus, very luckily, they are cross protective, and because they share so much of homology, so now, at least if we have a sheeppox vaccine or a goatpox vaccine, we can save our animals from either of these three diseases. So we should be extensively concentrating on use of vaccination programs. This is, at least for the time being, we can neglect. Pox viruses, we should really concentrate now extensively using our efforts, research, and orientation, and also possibly use on the tips of the earlier smallpox eradication, or at least in case of the veterinary practice, how we have eradicated successfully the Rhinovirus program. So we should really concentrate now on vaccines. Everyone today, particularly Dr. Ashok, he has been a really dynamic person, and I really appreciate his efforts of building a platform where like-minded people, including the farmers, for which we tried, which for his serious efforts, and are always open to help. And of course, this presentation was some of my own work and some then done by some of our PG students also. And I really thank and appreciate everyone that was closely associated in preparing this presentation, and as well as all present today who has given a patient here. Thank you so much. Dr. Ashok. Hello.
Hello.
Dr. Ashok. Sir.
Hello.
Sir.
Sir, please stop sharing, sir.
I've been fudging with the controls here. I couldn't find it. Okay, this is share. Stop sharing. Yeah, done.
Sir, actually here I am suffering with the power problem. No, I am operating with mobile, sir. Sir, there are few questions posted in the chat box. Can you please read out yourself, sir? Hello. Hello. Hello.
Can you hear my voice?
Yes, yes. Sir, everything completed? Huh?
Yes.
Hello.
Yes, sir.
Sir, you have answered all the questions, sir.
Answer. 2 questions I have already answered.
Sir, sir, here I am facing a power problem, sir. So I have connected with some other device.
Okay. Somebody, if you have any question, you can ask verbally or you can ask me.
Participants, if anyone is interested, just you can unmute yourself and ask questions. Sir, I think there are no more questions, sir.
Thank you so much.
Thank you, sir. Thank you very much.
Be available in the WhatsApp group. Even if you have some personal queries, you can always post them, and I will be happy, more than happy to reply to them personally.
Yes, sir. Thank you, sir. Thank you very much. As I am not able to attend from my regular computer, I am unable to share the slides, sir, remaining slides. So thank you all the participants and thank you the speaker, sir, and special guest. Our next webinar is scheduled on 6th November. I will be sharing the information through WhatsApp groups. Thank you all. Thank you once again. With this, we close this webinar. Thank you, sir.
Thanks to Josephina Thapa also.
Yes, sir. Thank you. Thank you.
Thank you. It was my privilege, really.
Thank you, sir.
Yeah.
Sir, sir.
Thank you, sir.
Thank you, madam. Thank you very much. close. All the participants kindly leave the meeting.